All right. We're going to talk about Mendel's scientific approach. I'm sorry that I think the previous little snippet had some of this information on it, but we'll go over it again real quick because I'm not 100% sure where you need to start. Mendel's scientific approach. Why did he use garden peas? Well, he did it because he had to, because that was his chore, that was his duty at the monastery. But garden pea was very good test organism because it's easy to grow. I can grow it. If I can grow it, anybody can grow it. It reproduces very quickly. You can have several generations during a growing season. If you use a greenhouse, you can get even more. They are self-fertilizing. Right, so most flowers have petals and a separate female part and separate male parts, right? And then some pollinator comes by, looks in here for some nectar, gets pollen from these male parts, the anthers, gets some stuck on his feet, and etc. And then he goes to another flower, and as he brushes up against the female part, the pollen gets stuck on the top. That's normal, regular flowers, okay? However, the pea has the female part, the male parts, and then their petals sort of overlap each other, okay? So they pretty much house all of the male and female portions of the flower inside the petals, and therefore the pollinators don't get to them, right? And so instead, the pollen that's produced from these anthers falls down and it gets stuck on the style of the female part, the pistil, and fertilizes itself. Okay, so it's self-fertilizing. So that self-fertilizing is important because you can control things by eliminating the pollen by cutting the stamens out, and then you can put whatever pollen for whatever trait that you're looking at and brush it onto the stigma of another flower, and you can control things. You can control pollination, right? So that's what self-fertilizing means, right? True bred hairs are those that when he observed them over generation and generation and generation and generation, they all produce the same phenotype, what it looks like. For instance, flower color. He looked at peas. Peas had purple and white flowers, and so the purple parents always, their offspring produce purple, and their offspring produce purple, and their offspring produce purple. All right. Likewise, the white flowers, they produced offspring that were white flowers, and those offspring produced white flowers. Okay. So those he called true bred parents, plants, all right? And then lastly is cross-fertilization, or cross-fertilizing. And this is a term, I just talked about how regular flowers are pollinated, and that's cross-fertilizing. But the term that we're going to be using is a little bit different term of cross-fertilizing. It's where you are in a controlled environment, where you can control certain variables of your test. And so he could take some of the different aspects, the phenotypes of these different parents, and under controlled circumstances, he could see what the fate of those phenotypes were over many generations. And I know I'm using the term phenotype. Phenotype is just what the trait looks like. Is it purple? Is it white? All right. So Mendel hypothesized or theorized that if you took a true-bred plant with a certain trait, like white flower color, and eliminated its source of pollen by cutting the stamens from that plant, the male parts, and then pollinating those plants with the pollen from a true-bred plant with the opposite version of the trait, purple flower, then he could track the fate of the trait, which in itself is flower color. The trait really is flower color. The purple and white are going to be called phenotypes. And so he could track that fate of that trait over generations. And if there were patterns to these traits' inheritance, those patterns could tell him something about the actual hereditary material itself. So there's some definitions before we get started. A gene is just a unit of information about a specific trait that are passed from parent to offspring. So a gene is the makeup, the genetic information that controls a specific trait. Diploid, I think we use this term in chapter 8, but diploid is a pair of chromosomes, all right, two sets of chromosomes. Allele is the molecular form of a gene, all right, and so an allele, normally there are two alleles that make up a trait, okay, and so we'll look at that here in a second. All right, so if we have an allele and we have two of them, because in normal situations there are two chromosomes, two genes, therefore two alleles, okay? If the alleles are the same for a certain trait, we call those homozygous, all right? If they are different, then they're called heterozygous. Hetero means different, homo means the same. Right. Dominant. Dominant is the allele that completely masks the expression of the other allele. Okay? And so that allele is normally designated by a capital letter. Right? So if we go back up here, we have two alleles. We have two that are dominant. Okay. Here, the heterozygous individual for gene for a trait is one dominant. Okay. The next word is recessive. That's the allele that, if they're in a heterozygous condition, that allele is masked, okay, by the dominant allele, right? And it's designated with a lowercase letter, all right? So if we look back at heterozygous, it had a dominant and a recessive allele. If we have two dominant alleles, the individual is called homozygous dominant. because homozygous means it has the same alleles. If it's homozygous recessive, it has two recessive alleles. Right? The term genotype, that's the particular genes that are present in an individual as represented by the alleles. So, for instance, a genotype could be homozygous recessive, it could be homozygous dominant, it could be heterozygous. Okay? That's the genotype. The phenotype is the actual observed traits of an individual. Are they purple? Are they white? Okay, that's the phenotype. When we take a look at these cross-fertilizing tests, we use some abbreviations for the parent generation. The parent generation in a genetic experiment is called the P generation. The first generation of offspring is F1. F1 stands for first filial generation. And then the second generation of offspring will be F2, or second filial generation of offspring. And so, this leads us into looking at specific traits, all right? Particular traits, and we're going to look at one trait at a time in the first series, and this is going to be called a mono means one, hybrid cross. So we're just looking at one trait during this particular experiment, all right, test. So Mendel took a true-bred pea plant for a trait like flower color and a specific flower color, white, and he cross-fertilized or crossed with it a true-bred pea plant that had the different expression of the trait, which was purple. Okay? The resulting offspring in the first generation were all the dominant phenotype for that trait. So they were all purple. When he crossed the two F1 offspring, if he took one of these, purple, okay, and crossed it with another one of these, purple, then for every 4 individuals in the next generation, he got 3 that expressed the dominant phenotype. So he got 3 that were purple, but then he got 1 that was expressing the recessive phenotype, white. Right? We're going to go over this here in a second. So his conclusions were that purple was the dominant allele, white was the recessive allele. He started with true-bred parents, so the purple parent must be homozygous dominant for the trait, and then the white parent must be homozygous recessive for that trait. Each parent would donate 1 allele to the offspring in the first generation, and therefore the only allele that the purple parent could donate would be the dominant allele, and the only allele that the white parent could donate would be the recessive allele. So the resulting offspring would have a heterozygous genotype, right? 1 dominant and 1 recessive. Since the purple flower color is dominant, all the offspring in that first generation express the purple phenotype. When he took 2 F1 heterozygous offspring and crossed them, each parent then could donate 2 alleles, 1 dominant and 1 recessive. Therefore, for every 4 F2 offspring produced, 1 individual would be homozygous dominant, 2 individuals would be heterozygous, and 1 individual would be homozygous recessive. The genotypic ratio, all right, this is the ratio of what out of every 4 individuals in a monohybrid cross in the F2 generation, 1 will be homozygous dominant, 2 will be heterozygous, 1 will be homozygous recessive. So that's where 1 to 2 to 1 genotypic ratio can be found. 3 would show the purple flower color, and 1 would just show the white. So the phenotypic ratio would be 3 purple, 1 white. So the phenotypic ratio is always 3 to 1. Since fertilization is a chance event, Mendel could use probability to help him predict the possible outcomes of these crosses. So that's how he came to these conclusions. Right? So, we will look at the Punnett square here in a second. All right, so we have our parent generation. That work? All right, that's good. We have our parent generation. And we start with a purple purebred pea, all right? And we cross that with a purebred white flower pea, okay? If they're true bred, they are going to be homozygous dominant for purple. And for white, it's going to be homozygous recessive. Now, I use P. You can use any letter to signify these alleles, all right? I'm just going to use P, right? But you do need to use a capital letter for the dominant allele and a lowercase letter for a recessive allele. So in the F1 generation, let's say this is the male and this is the female, all right? So in the gametes, in the pollen, okay, the only allele that the male can produce is a big P, right? It's the only one that it can transfer to the next offspring. Likewise, the only one that the white can provide is a little p, right? So when you cross them, you fertilize them, in the F1 generation, we get a dominant allele and a recessive allele, all right? And so, this individual is going to be purple because the dominant allele, purple, masks the smaller recessive allele, white. Okay. Now, if we took another individual from this same generation and we crossed them in F1 generation to see what we get in the F2 generation. Now this individual can provide a dominant allele and a recessive allele in its gametes. Likewise, this individual can provide the same gametes, all right? Same alleles in the gametes. So there are several possibilities now. If you could have one big P fertilize another big P, or this big P could go over and fertilize that little P, right? Likewise, this little P can go over and fertilize a big P. I'm going to make the big P first. or this little p can go over and fertilize the little p. Okay? So 4 individuals, 4 possibilities, all right? This individual is going to be purple. This one is 2, and this one is 2. This one is going to be white. So if we look at the phenotypes, there are 1, 2, 3, 3 purples, and we have just 1 white. So the phenotypic ratio of a monohybrid cross is always 3 to 1. It doesn't matter what trait you're going to be looking at, whether it's tall plants versus short plants, all right? Thorn versus thornless, all right? It's always going to be 3 to 1. The genotypic ratio all right, has 1 that's homozygous dominant. We have 1, 2 that are heterozygous. And then we have 1 that is homozygous recessive. So the genotypic ratio is always going to be 1 to 2 to 1. All right? Doesn't matter what trait you're looking at. It's going to be the same. Now you can use a thing called the Punnett square. The Punnett square is just a way to help you keep track of the gametes and the alleles that can go in those gametes. For a monohybrid cross, we really don't have to worry about Punnett square too much, because you can just memorize or just know that the genotypic ratio is always going to be 1 to 2 to 1, and the phenotypic ratio is always going to be 3 to 1. But to use it, what you do is you took your true bred parents, you got these heterozygous individuals in the first generation. So you take these 2 individuals and line up what alleles that they can produce. So this one can give a big P and a little p. All right? Over here, this one can give a big P and a little p. And so what you do is just match the letters together, and you come up with your genotypes, right? The genotypic ratio now is still going to be 1 to 2 to 1, all right? The phenotypic ratio, this is purple, this one's purple, this one's purple, 3, this one's white, 1, okay? So that's always going to be the same. It does not matter what trait you're looking at. The phenotypic ratio and the genotypic ratio are always going to be the same. And the genotypic ratio is 1 to 2 to 1. The phenotypic ratio is 3 to 1. You can test cross. A test cross is just a way to determine whether the dominant parent is going to be true bred or is going to be heterozygous, okay? Because if it's heterozygous, it can provide the recessive allele, and if it does, then in the first generation, white plants will show up, not all purple like before. And so that's a way to determine. You can look at genes and expressions of those genes. You can determine parenthood. Or you can look at carriers of diseases later on. We're going to talk about in chapter 9 about that too. So that's called test cross. So that is just one gene at a time, all right?